Okay, so once we have our light focused on our retina, our photoreceptors are going to start to come into play. So we'll talk just briefly about the functional anatomy of both the rod and cones, which are our photoreceptor cells, and then we'll talk a little bit about the chemistry and the response of the visual pigments to light. And then we'll also talk a little bit about the photoreceptor activation, phototransduction, and how we process the information that's coming into the retina. I do want to point out you just need to have a general understanding of what's going on. None of the specifics, even if I have them listed here, are going to be on your exam. It's going to be a very brief section, again, on the special senses. So we're talking about our photoreceptor cells, which are our rods and our cones. So rods and cones, they do have different thresholds for activation. So rods are really, really sensitive. They respond to very dim light, making them best suited for night vision and peripheral vision. When we're talking about cones, these need bright light for activation, but they react much more quickly. And cones have one of three different types of pigments that furnish a vividly colored view of what we see. But our rods contain a single kind of visual pigment, so their inputs are only perceived in gray tones. They also differ in their wiring to the retinal neurons. So rods participate in converging pathways, and because of that, the rod effects are summated and considered collectively, resulting in vision that is fuzzy and indistinct. When we're talking about cones, each cone in the fovea has a straight-through pathway via its own personal bipolar soul to the ganglion cell. So each foveal cone has its own direct line to the visual centers that are higher up. And this allows for detailed, high acuity or high resolution views of very small areas of the visual field. And remember that rods are absent from the fovea and cones do not respond to low intensity light. So we see dimly lit objects best when we actually don't look at them directly, and we recognize them best when they move. So pretty much what you need to know for our photoreceptor cells, which are our rods and the cones, be able to compare and contrast them. This table pretty much has everything highlighted for you that you'll be responsible for on your exam. The next thing we're going to talk about is phototransduction. And phototransduction is the process by which light energy is converted into a graded receptor potential, kind of like what we saw occurring in the dendrites and the soma of a neuron. So it's going to begin when a visual pigment captures a photon of light. So the visual pigment of rods is a deep purple colored pigment called rhodopsin. And when we're talking about rhodopsin, it's arranged in the rod's outer segment. And there's 3 steps of rhodopsin formation and breakdown. The 3 steps are pigment synthesis, pigment bleaching, and pigment regeneration. We have a similar process that also occurs in cones, but we're talking about different opsins, and cones require more intense light, as mentioned previously. Okay, step 1, which is pigment synthesis. Our rhodopsin forms and accumulates in the dark. Vitamin A is going to be oxidized to its 11-cis retinol form and then combined with opsin to form rhodopsin. Again, that's a lot of information, but what you need to know for phototransduction steps are given to you here. The next step, or step 2, is pigment bleaching. So when our rhodopsin absorbs the light, the retinol is going to change shape to its all-trans isomer, which is, again, too much information, but I'm just letting you know, which is going to allow the surrounding protein to really quickly relax, like similar to like an uncoiling spring. And when it does this, it's going to be in what we refer to as its light-activated form. Eventually, the retinol-opsin combination is going to break down, which allows for retinol and the opsin to separate. The breakdown of rhodopsin to retinol and opsin is known as bleaching of the pigment. So that's where this step gets its name. And the final step is going to be pigment regeneration. Once the light struck all of those transretinal, it's going to detach it from the opsin. So we have enzymes within the pigmented epithelium that are going to reconvert it to its 11-cis isomer, and then the retinal basically is going to heads back to the photoreceptor cells' outer segments where it began. So rhodopsin is going to be regenerated when the 11-cis retinal is going to be rejoined to the opsin. The breakdown and the regeneration of visual pigments in these cones is essentially the same as rhodopsin, like I mentioned, but we just have cones that are about 100 times less sensitive than rods, which means that it takes a higher intensity or a brighter light to actually activate the cones. So this is pretty much an outline of what we just talked about when we talked about phototransduction and the 3 steps. So when we talk more about light transduction reactions, the light-activated rhodopsin is going to activate G-protein transducin. Transducin, in turn, is going to activate PDE, which is a phosphodiesterase, and this is the enzyme that's actually going to break down cyclic GMP or cGMP. So if we're in the dark, cGMP is going to bind to cation channels in the outer segments of the photoreceptor cells and basically hold them open. And this is going to allow for sodium and calcium to come into the cell. If we're having positively charged ions coming into the cell, this is going to cause depolarization of the cell. And this is going to form what we call the dark potential, which is about negative 40 millivolts. If we're in the light, our PDE or phosphodiesterase enzyme is going to break down cyclic GMP and the cation channel is going to close. So we don't have sodium and we don't have calcium entering into the cell anymore. And this is going to cause the cell to hyperpolarize to about negative 70 millivolts. So this is showing you the events of phototransduction. When we have a portion of the photoreceptor disc membrane shown here. So the G protein conversion of GTP to GGP is not here because we're trying to keep this pretty simple. So we do have our CR channels that are gated by cyclic GMP or cGMP, and those are in the membrane. And we also see our visual pigments in the plasma membrane as well. So you can kind of walk through these different steps of what's going on. We have our retinal that's going to absorb light and changes its shape, and we have our visual pigment that activates. We go through different changes, cis and trans. This is just referring to the location of the hydrogens, whether we're on the same side of a bond or opposite side. So cis means the same and trans means opposite. So that's not as important. The big picture is what's important, which this image is showing you. The next step is where we have the visual pigment activate transducin. Transducin is a G protein. So our G protein is going to translocate across the plasma membrane until it binds PDE, which is the enzyme, the phosphodiesterase. Once we have it there, it's going to activate phosphodiesterase, and then our phosphodiesterase can convert cyclic GMP into GMP, causing the cyclic GMP levels to fall. So as cyclic GMP levels fall,